In this chapter, we'll start to look at some of the biotechnologies that are involved in DNA analysis. This is in chapter 19 of your textbook, and what I plan to do is to separate this out into relevant parts as we talk about those aspects in lecture. So we will just focus on a few items from this chapter at this point, things specifically that involve DNA structure, which we've just talked about. So first of all, I'd like to talk about restriction enzymes. Restriction enzymes are enzymes that naturally occur in bacteria. They're a mechanism of defense for bacteria against DNA that they may take up from their environment. So, restriction enzymes specifically cut DNA, make a double-stranded DNA cut at a particular sequence, and that sequence is called the recognition sequence. There are many different restriction enzymes that have been isolated from a lot of different species of bacteria, and conveniently we can just order these from a company. So there are a lot of examples of restriction enzymes. So ALU1 specifically binds to the DNA sequence AGCT, and it makes a cut right in the middle of those bases. Most restriction enzymes actually recognize a palindromic DNA sequence. And so you'll notice that 5' to 3' on one strand is AGCT, 5' to 3' on the other strand is also AGCT. That's a palindrome. Some enzymes have a recognition sequence of only 4 bases. Others have a longer recognition sequence. So, for example, BAMH1 has a recognition sequence of 6 bases, and this one cuts slightly differently. So it cuts both of the strands of DNA, but it actually is a little bit staggered. And so we end up with what's called a sticky end, where you have this staggered cut that leads to a small region that is single-stranded at the end of that DNA strand. We will be using restriction enzymes in lab in the second lab project, the PGLO project. So you'll get some practical experience with these. The next thing I'd like to talk about is how we actually visualize DNA when we're working with it in the laboratory. We use something called agarose gel electrophoresis. And what this does is it separates the DNA by size. And so the agarose acts sort of like a sieve, and the DNA will move through that agarose gel. This is all based on the fact that DNA is actually negatively charged, and so it's going to move toward the positive cathode here. So as we run the current through the gel, the DNA will load it up here at the minus end, and it will migrate through the gel toward the positive cathode. The larger pieces move much more slowly, okay, and the smaller pieces will move much more rapidly. And so at the end, after you have separated those pieces of DNA, the smallest pieces are at the bottom and the largest pieces are at the top. One thing that I want to point out is that you need a lot of DNA to actually be able to visualize it on a gel. So you need many, many copies in order to see it. The other thing is that we use a dye that binds to DNA, and this will actually show the position of bands of DNA, so where we have discrete sizes of DNA. The last technique I'd like to talk about is a technique for locating a particular DNA sequence within an entire genome. And this technique is called a southern blot. And first I just want to introduce one aspect of this technique, which is called probing. So we use the complementary nature of DNA in order to locate that particular known sequence that we'd like to find. And so it turns out that when you isolate DNA, if you heat it up, you can actually separate these two strands. So here's the double-stranded DNA, and if you boil that DNA, it actually separates into these two single strands. And then you can use something like a radioactively labeled probe, as it's called, to complementary base pair with that particular sequence to locate that particular piece of DNA with that complementary sequence. So we use probing in a technique called a southern blot, and this uses both restriction enzymes and gel electrophoresis. So, a southern blot, again, is to determine whether or not a particular sequence is present in your sample. And so, in this technique, there's a couple of different steps. So, you would start off with chromosomal DNA, and usually people then take that DNA and cut it up with possibly a variety of restriction enzymes. So, that is shown with these scissors here. Following that restriction enzyme digest, we would run that DNA, separate it by size on a gel electrophoresis, and now we have lots of DNA bands at different sizes, with the largest pieces at the top and the smallest ones at the bottom. Following gel electrophoresis, we would then transfer that DNA from the gel onto a membrane. And so this would use horizontal application of a current to move that DNA out of the gel and onto that membrane just underneath it. Finally, we would use a radioactive probe to locate the position at which that sequence of interest lies. And so here we see that radioactive probe bound to this particular band, and that would tell us that that particular sequence was present in our genomic DNA sample.